It's an honor to be here to discuss our research on overcoming osimertinib resistance in EGFR mutant non-small cell lung cancer through inhibition of pyruvate dehydrogenase kinase. I will provide some background information. Lung cancer is the second most common cancer, and about 80 to 85% of the lung cancer is non-small cell lung cancer. Non-small cell lung cancer patients usually undergo the molecular profiling to determine the best treatment options, and EGFR is identified as one of the most common mutations. In normal cells, EGFR, epidermal growth factor receptor, is activated by binding to its ligand and leading to receptor dimerization and autophosphorylation in its intracellular kinase domain and activation of the downstream signals. It can modulate the cell growth, proliferation, and survival. However, the mutations around the ATP binding pocket, including the exon 19 deletion and L858R mutations, can lead the ligand-independent autophosphorylation, also can lead the downstream signal hyperactivation. It can promote cancer cell survival, proliferation, migration, and invasion. And to address this challenge, EGFR tyrosine kinase inhibitor, EGFR TKI, were developed to competitively binding to the ATP binding site of the EGFR, can effectively blocking the receptor's kinase activity. And osimertinib is a third-generation EGFR TKI that shows high selectivity in both EGFR activating mutation patients and the secondary acquired EGFR T790M mutation patients. However, the resistance to the osimertinib usually occurs within 10 months, and the majority of the cases is mediated by EGFR C797S mutation, and this study aims to overcome this resistance. According to two previous studies, combined targeting of the EGFR and PDK can enhance the therapeutic efficacy. Thus, in our study, we are focused on targeting the pyruvate dehydrogenase kinase. PDK is a mitochondrial kinase enzyme that can inhibit the activity of the PDC through phosphorylation 8. And PDK1 is usually overexpressed in cancer. Before the study, we hypothesized that by PDK inhibition, we can overcome the osimertinib resistance mediated by the EGFR C797S mutation. And this study mainly has 4 parts: in vitro study, patient sample, allograft, and xenograft. First of all, we have constructed the EGFR mutant cell lines. We have selected the A549 cells as the primary cell lines and knock out the endogenous EGFR expression using the CRISPR-Cas9, then re-express the EGFR wild type, EGFR single, double, or triple mutation to mimic the common clinical situation. As shown in figure C and D, we can find that all the cells with the EGFR triple mutation develops the resistance to the osimertinib. Furthermore, we have stably overexpressed the EGFR double mutation, which are sensitive to the osimertinib, and EGFR triple mutation, which are resistant to the osimertinib in H292 cells, PC9 cells, and H1975 cells. And all the cells harboring the EGFR triple mutation acquired the ability to resistance to the osimertinib. Next, we have compared the expressions of PDKs in the EGFR mutant cell lines. And we find that the expressions of the PDK1 is significantly increased in EGFR triple mutant cells, and also the 3 phosphorylation sites were increased in EGFR cells harboring the EGFR C797S mutation. Next, we have a collaboration with the Sun Yat-sen University Cancer Center to analyze the patient sample from lung tissue. And we find that the osimertinib resistant patients has higher levels of the phospho-PDHA and also increased the expressions of the PDK1. Then we use the cell lines to compare the glycolysis ratio by measuring the glucose uptake, lactate production, and oxygen consumption rate. And we find that cells with EGFR triple mutation has increased the levels of glucose uptake, lactate production, and decreased the levels of the oxygen consumption rates. And all these results indicated that the cells with EGFR triple mutant has a higher levels of the glycolysis. Then we explore the correlation between the EGFR mutation and the PDK1 upregulation. We have analyzed the expressions of the EGFR signals, and we find that the EGFR signal, including the phospho levels of the EGFR, AKT, ERK, and STAT3, were increased in EGFR triple mutant cells compared to the double mutant cells. And also the upstream of the PDK1, HIF1 alpha, was also increased. And next, to tell which pathway increased the expressions of the PDK1, we have used the 3 inhibitors: PI3K inhibitor, MEK inhibitor, and JAK inhibitor. And we find that only using the PI3K inhibitor, the expressions of the HIF1 alpha and the PDK1 were decreased. But using the HIF1 alpha inhibitor, YC1, can decrease the both expressions of the PDK1 and the PDK3. Then we have treated the cells with the osimertinib, and we can find that the cells with the EGFR triple mutant has less decreased the levels of the phospho-EGFR, phospho-AKT, and HIF1 alpha and PDK1. And all these results indicated that the EGFR C797S mutation increased the PDK1 expression through the pathway of the EGFR, AKT, and HIF1 alpha axis. Next, we try to overcoming the osimertinib resistance by PDK inhibitor. We have selected the 2 PDK inhibitors through our previous study, and DCA is one of the most investigated PDK inhibitor, and the lilamin shows the high selectivity to the cancer. And we can find that by combination of the osimertinib with the PDK inhibitors or upstream of the PDK inhibitors can overcome the osimertinib resistance in the MTT assay and Western blot and FACS analysis. Then we have conducted an allograft model. We stably overexpress the EGFR double mutation labeled with BFP and EGFR triple mutation labeled with GFP in LLC cells. Then we mix them into 1:1 ratio to mimic the tumor heterogeneity, then inject it to the mouse. Then we treated the mouse using the osimertinib alone or osimertinib combination with the PTK inhibitor lilamin. And we can find that by combination of the osimertinib and the lilamin can effectively inhibit the growth of the tumor in allograft. At last, we try to overcome the osimertinib resistance using the CRISPR. First, we have conducted the in vitro study. We knock out the expressions of the PTK1 using the CRISPR-Cas9, and we can find that all the PTK1 knockout cells has the increased sensitivity to the osimertinib. Finally, we have doing a xenograft study, and we find that using the CRISPR-Cas9 by knock out the expressions of the PTK1, the tumor growth can significantly inhibited. In conclusion, we find that the EGFR C797S mutation increased the PTK1 expression through the PI3K AKT and HIF1 alpha axis, and by inhibition of the PTK1 can overcome the osimertinib resistance mediated by EGFR C797S mutation. However, our study also has many limitations, and still some of the study is going on. And I want to thank to all my colleagues, and thanks for your listening.